We had to juggle a few things. And it is a real pleasure to introduce Dr. Anita Bandrowski to speak today in our High Fidelity seminar series. I've known Anita for a few years. We both have a keen interest in the importance of standards and in reproducibility. And as you'll hear today, Anita's resource identification initiative is really, I think, one of the most interesting and innovative and potentially impactful that really has come about since the discussion about reproducibility has come to prominence. By way of introduction, Anita trained as a bench neurophysiologist working to elucidate physiological mechanisms of learning in epilepsy. After her postdoc, Anita worked in data, starting with the annotation of the human genome for Celera, which actually there are some people here at Frederick who have come through Celera. Anita then moved to neuroinformatics with an award from the Neuroscience Information Framework by the NIH's Blueprint for Neuroscience. And the goal of this project was to create a comprehensive list of databases for neuroscience and to federate a search across as many of these databases as possible. And that framework really grew to the most comprehensive search system for neuroscience data on the web. And this broad overview of the data landscape also highlighted the dearth of reagent information, and in particular, how reagents and tools are cited in the scientific literature. If you can't, follow the reagents and the lot numbers, pretty hard to reproduce a given experiment. And so to address reagent underreporting issues, Anita serves as the lead for the Research Resource Identification, RRID, initiative that I mentioned a minute ago. And this group is dedicated to transforming, as you'll hear today, scholarly communication, which has recently become, and it's recently become a nonprofit organization. RRIDs are unique identifiers for key biological resources aggregated from the group, from community databases, and requested from authors in participating journals. As I said, I think it's a really powerful and important innovation in combating irreproducibility. So the seminar title today is RRIDs for Research Resources. Why do we need a number to make resources fair? Capital F in fair. Anita, it's all yours. Thank you again for coming virtually.
Thank you so much, Len. That's a really nice introduction. So let me try to share. And I forgot what the title was supposed to be, so here's what I thought it was. But that's okay. I think we're about as good as we're going to get. Can everybody see my screen? And please actually stop me. I don't think this is as formal a talk as, it doesn't need to be a truly formal talk. We can just stop this whenever. Okay, so this is, again, and I have an appointment at the Department of Neuroscience at UCSD, and then I have a conflict of interest statement with SciCrunch. This is a company supported by an SBIR. I will point out when that becomes an issue, a potential issue. Okay, so the NIH circa 2008 had as basically part of the Neuroscience Information Framework contract to UCSD, in which I participated in the fulfillment of that contract, had basically said something to the extent of, and this is not the actual contract language, but people are essentially wasting a lot of time looking for antibodies. Why can't you help them by just creating an algorithm that can do that for them? It's just really, this is such a mundane, easy, trivial task. Why not just, get a robot to do it for them? So what we did is we started to actually work with the Journal of Neuroscience. It's our big society journal. And what we ended up doing is licensing out a year of the journal from them so that we could text mine all of that content. And then, but we also had to start working on actually trying to find these things in papers. And, you know, we had done this, all of us had done this long ago in grad school. And this is an example of the kind of thing that we flagged. We said, hey, look, this looks like an antibody, mouse anti-flag from Sigma. That's great. So we tried to highlight that. And then we tried to do the second step, which is, oh, there we go, to try and look for that antibody in the catalog. And what we found is usually not just one, but actually multiple antibodies that matched a particular description that the authors gave us. And in fact, this was very, very common. So we went back to the editorial board, the full editorial board of the Journal of Neuroscience, and we sat around and we said, look, here are our findings. You know, you guys are not having more than about 10% of catalog numbers. And none of the papers we looked at actually had a lot number associated with any of the antibody citations. We've got to do something about this. And there were murmurs all around the room, and they basically said, yeah, this is terrible, they're terrible, 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 terrible. And then we came back a year later and nothing had changed. So we thought, that's interesting. The other thing that really kills me here is that here's another example of something like this for a mouse. So this mouse was purchased from Jackson Laboratories, right? And this is how the authors describe it. The problem is this mouse also does not exist in the Jackson Labs catalog. So there's actually no mouse with this description. But the author actually knows exactly which mouse they used because they have the strain number. And so, you know, for this particular paper, the author got back to me within a couple of hours just to figure out, you know, just to let me know, you know, what the mouse is. And this means that there are good notes in these laboratories. It's not a problem of note taking, but it is a problem of actually putting this information into the paper. Our colleagues from OHSU had actually quantified this to a great extent. And this is, again, this is no robots. This is only a single curator who went through several hundred papers, looked at a bunch of antibodies, cell lines, constructs, knockdown reagents, and organisms. So he looked at it across different impact factors and different fields in biology. But overall, this is what the picture looks like. Half the antibodies could not be tracked down using a very loose criteria. And, you know, half the cell lines couldn't be tracked down. Organisms did a little bit better, but not in neuroscience. So this is a very significant problem. And so really, we could not answer which antibodies, which reagents are being used in this paper. So what that means is that paper cannot be reproduced for a really stupid reason, which is sitting in a laboratory notebook somewhere. And then, of course, we can't figure out which other papers, which other people, which other labs are actually using this particular reagent because the information is not in the paper. So what we also didn't know at the time that we started all of this is how many antibodies are even out there for people to use, right? And because we didn't know that, we actually built the antibodyregistry.org. We gathered data from about 250 vendors and started working really closely with the Journal of Comparative Neurology to define which of these antibodies exactly are being used in which of the Journal of Comparative Neurology papers. And this is really how the antibody registry started and started to become useful. So we gave every antibody an identifier. Those later became the RRIDs, the Research Resource Identifiers. But again, how do we get these IDs into papers where they could actually be useful? So we knew that just telling a journal editorial board would actually have essentially no effect. So in 2012, we actually had hosted a meeting at the Commander's Palace at the Society for Neuroscience meeting. with 25 journal editors-in-chief. All of the big journals were there, and that was wonderful. So we learned a few things during that meeting. First of all, we were able to convince these journals of the first important part of all of this, which is that, you know, this reagent identification is a problem, and it is a problem in their journals. So that was the first thing, and that was a very important thing. We also learned that no matter how hoity-toity, almost no one refuses a meeting at the Commander's Palace because it's really good food. So that was the other thing we learned. But then in 2013, NIDA actually sponsored a 2-day workshop with these same 25 journal editors-in-chief, which was extraordinarily helpful in nailing down what a pilot project might look like. And then there's a meeting in here that didn't get put onto these slides, but essentially at the following year's Society for Neuroscience meeting, we had actually agreed on a start time for the pilot project. And this was supposed to be a 3-month pilot project with these 25 journals who would include, you know, these RRIDs in the journal somewhere. So some of them put it into their instructions to authors, some of them sent out letters, some of them worked with authors directly to get these things into the papers themselves. So basically in about 2 months over the holidays, we needed to aggregate all of the information that was needed, not just for antibodies, but also for software tools, which was agreed upon by the journals, as well as about 25 different model organisms, put up a website that could, you know, that could be used by authors easily. For that, we needed a help desk. Oh, and by the way, we didn't have any additional funding to do this. So that was fine. We were actually able to do that. So 2 of the journals actually started on time. The rest of them started between 1 and about 18 months later. But on the positive side, we are currently in the 7th year of this 3-month pilot. And our latest, you know, big journals are now include Nature, Science, Cell, eLife, who are all on board, as well as a lot of the society journals, including, of course, our society, which is the Society for Neuroscience. Okay, so what are these RRIDs? They literally break down into the full syntax, which is the company name, where is the place that reagent or research resource exists, what is the catalog number of this thing. We also suggest a lot number, and that typically is after this. And then there's the RRID, the identifier from the authority for that particular item. So who are these authorities? Well, I've talked a little bit about the antibody registry. That's actually an authority that we run. But there are lots of other authorities. Biosamples is an NCBI joint project with the European Informatics Institute, and that provides RRIDs for biosamples. Cellosaurus provides RRIDs for cell lines. And then there is a ton of organism providers. You probably know many of these. You probably don't know them all. I don't think I know them all, even though I aggregated them. But places like MMRRC and Jackson Laboratories are just some of the authorities for mice. Of course, the mods, MGI, actually take care of a lot of the mouse requests. There are worms, flies, rats, pigs, and others. Adgene currently serves as our authority for plasmids. So these things, all of these are authorities. So the authorities say, here's an item that you can essentially buy, obtain in some way, and use in your research. And here's a little bit of information about this thing. And those are the kinds of things that the kinds of information that these authorities have. But RRIDs also have something else in them. So because people are using the RRIDs now in the literature, we can actually mine the scientific literature for the mentions of these RRIDs. And so we're starting to create some very interesting artificial intelligence bots that are going through and mining this out of the literature in terms of what the people have actually said about some of these things. And of course, at least we have a little bot that actually used that is used by curators to mine these things also, or pull these things from the literature that at least tells you which one of these is being used. And then there's a bunch of other projects. Encode is a publicly funded initiative. They have a lot of data about how antibodies are performing, for example. A lot of core facilities have some of that data. Human Protein Atlas has some of that data. ICLAC is the International Cell Line Authentication Committee. I think a lot of you know about this, but they have information primarily about which cell lines are actually compromised in some way. But all of that goes into a single portal, and that is a very useful thing. This is what it looks like. So here is a search that I'm, we're basically asking, so the journals are asking for the author to go ahead and go to a website to find all of their RRIDs. So the author goes in, searches for whatever they're looking for. In this case, this is a core facility. They are then asked to cite this. There's 2 places where that information lives on this, and the author has to cite this text, put it into the manuscript, and then the paper is actually published. So here's just a search for one of the Jackson mice, and there are about 900 papers that have been published with this mouse. The RRID portal actually includes a lot of different things. So it includes antibodies, organisms. There's about 500,000 from over 25 centers right now, actually. There's over 100,000 cell lines that are listed, a lot of plasmids, about 80,000. And then there's a lot of statistical tools, core facilities, et cetera. And that accounts for another 14 to 15,000. So that's roughly what the workflow is for the authors. And now RRIDs have also become part of the JATS standard. This is the Journal Article Tagging Suite. The version, which is version 1.2, actually includes this resource wrap tag. So this is now able to be created as a tag, as a specific tag by journals. So we will not be the only ones that are mining these things out at some point once this is adopted. And of course, RRIDs are part of multiple checklists, including STAR methods from CellPress, and the most recent MDAR, which is the Materials Design Analysis Reporting Checklist for authors. And you can see that actually the first item in this MDAR checklist is antibodies. The second item is cell lines. And of course, that checklist goes through, hey, go find a catalog number and an RRID. So that is true also for organisms. Again, this is required currently by science from all authors. Okay, lots of different places, including commercial vendors, like this one is BioLegend, but Thermo Fisher and many others actually also have this RRID as part of their material data sheets and web pages. MMROC actually takes the copying and pasting to a whole new level. You just have to click one button and you have copied the RRID citation to the clipboard, which is great. And of course, Cellosaurus, this is out of Switzerland, also has the how to cite information for all RRIDs. A few grants have RRIDs specifically listed as, if they're expecting to create research resources, they are expected to actually register those resources to get the RRIDs. So that is expected in several grants. And as I've mentioned before, we have some nice robots that actually help us out. So this is an example of a paper that has a couple of antibodies listed using the RRID. These RRIDs are actually read by a little robot called Cybot. And the information from the database is actually pulled here to the side of the article. Then the curator can make can decide whether that is the right antibody and can validate that. And once the curator has done that, then there's another robot that actually tweets about the fact that this happened, and that whole process can take as little time as about 2 days from when a paper with an RRID is published. So that's kind of our shortest possible time to tweet from finding the paper. So in this case, RRID Robot here is tweeting that this particular, American Chemical Society paper is actually using RRIDs for their antibodies. So that's kind of fun. So, question, did it work? Did we actually get anything that was successful? And we certainly are in lots and lots of different journals. So in that sense, it definitely worked. The STAR method section is now very prominent. It's required in all self-press journals. eNeuro and Journal of Neuroscience are big proponents. Other society journals, eLife, again, big proponents. This is what it looks like, right, in a particular paper. And so interestingly, now, instead of being confused and having to email an author about a particular Jackson Labs mouse, you can simply click on the RRID, and this information is typeset into the paper by the journal. This is not the author doing this. The author just provides the identifier, and the journal actually provides the link. And through a resolving service, we actually get back to, magically, the actual web page from Jackson Laboratories that talks about the mouse and a place you can actually order that mouse if that is what you really want to do, or find out a lot more information about this mouse. So in that sense, it worked. When we looked at in 2015, and this is only at the first 100 papers, that we're using RRIDs, we looked in the essentially at the percent identifiability. So remember back to that Vasilevsky paper, 50% of antibodies were identifiable before, in 2013. We were able to confirm that with the issue of the journal right before we implemented RRIDs. And so you can see roughly 50% of the antibody mentions are considered findable or identifiable. But in the very next issue, we actually were able to find that 90% of those were actually findable. So, going between 50 and 90 is a pretty big deal. And that was also true for tools and for organisms. So that was great. Some journals joined because of this. But remember that information that I told you from ICLAC and other places, where the information about a particular reagent having a particular problem is actually listed on the RRID website. So in this case, here I'm searching for the 15C4 cell line, and that is a problematic cell line. This one has actually been shown to be a HeLa derivative. This is verified as being contaminated by the ICLAC community. And so what happened is, we believe now very strongly with some substantial evidence that the authors that are actually looking for these RRIDs also see comments and problems that they're being warned about here. And the reason that I say that is because when we went out, and again, this is in 2019, we looked at all of the available papers from PubMed Central at that point that actually used a cell line. And we found that out of that subset of papers, which is 150,000 in 2019, we found that about 16% of those papers had used at least one problematic cell line. So this is huge and a huge problem. But when we looked at the 634 papers which were published that actually had used an RRID, the percentage of those cell lines that were problematic in some way was only 5%. So this is a huge reduction. This was very, very significant. And basically, showing that people can actually see this. So this was actually, there were 50 cell lines that were, or 51, I can't remember exact numbers, but there was about 50 cell lines. We looked at these very closely to figure out what people were using exactly. And there was only one of these that was actually truly contaminated, where the authors could know about that contamination and still chose to put the RRID in and publish that paper. Everyone else seems to have, all the rest of these cell lines are largely the problematic cell lines, things that are misidentified, also go on that list. We didn't make that differentiation here, but we didn't know this was way more, obviously, cell lines to look at. But here we could really disentangle these and really found, only one case where there was an RRID for a truly contaminated cell line. So when AACR, the American Association for Cancer Research, saw these results, they actually joined the RRID initiative very strongly, and now requesting, and I'll show you some more things about what they're starting to do now. Okay, so the other point about did this work is really also being made here. So we just published this last year. This is, again, another large swath of an analysis. This analysis is based on 316,000 antibodies from 682 different journals. Again, this was published in 2020. And this is that identifiability percentage. Again, I should have had my label here. I apologize for that. But you can see that in 97, 98, 2000, what we're looking at here is, and this algorithm is not looking for identifiability quite in the same way, because robots are not going to be able to tell very well whether, in 97, there's a catalog that corresponds to a particular name of an antibody. So this is really looking only for catalog numbers. So if you look at catalog numbers, about 10% of the journals had, of the antibodies that were cited in journals, had a catalog number associated, which is consistent with what we had found for the Journal of Neuroscience, somewhere around here. And that we find across all journals that were represented in the open access subset of PubMed Central. And this blue line shows that it's actually, the situation is getting better. Certainly, it's getting better over this 20-year period, going from, on average 10 to on average 20% of the literature has a catalog number associated. Woohoo! But here's where we started our pilot at this line. So in 2014, we started with those 25 journals. Again, many of them lagged behind substantially, but you can see a couple of those journals here that really started with us and started helping us. And they flipped from this kind of average rate of antibody identification to the 90% rate. And when they do that, they're actually starting to bring up the average. So when we take the average antibody, now the average antibody is, being cited using a catalog number. A lot of these do have RRIDs, and that is a substantial shift from about, 20% to about 45%, which I have to say is a huge overall shift in the literature. There's still a long way to go to bring the kind of stragglers along, like PLOS and non-co-target, but we certainly do have a big shift. And in fact, there's a little bit of a good news here. Even the stragglers are kind of coming along. So it seems like we've solved the entire reagent problem, right? Well, not yet. Certainly not yet. It turns out that, with these journals that have been asking for RRIDs, it takes a lot of time on the part of the journals to be able to do this. So we thought about this a lot, and we started to create tools to help journals actually improve RRIDs or RRID enforcement. So we reached out to a lot of commercial and nonprofit journals, and we started asking them questions. We applied for some SBIR funding, founded a company. So this is my conflict of interest statement. And we created this tool, which is intended specifically to be sold to publishers So it's currently being used in production at the AACR and also Microbiology Society. So I'm just pointing that out as a potential conflict. So what does this tool do? Well, what it does is it reads the methods section. And when it reads that methods section, it also looks for a whole bunch of things that are associated with one of the guidelines, including, of course, the RRID, including the RRID guideline. And then what it does is it says, hey, I'm expecting a certain number of these things, and I am not scoring or I'm not finding these. So just a little blow up. So in addition to finding a statement about antibodies and being able to find those antibodies or cell lines in the catalogs, it also asks for things like institutional review board statements, consent statements, and these 1 through 5 are the different kinds of guidelines. So you might be interested in the Landis 2013. This is the NIH guideline. Here are the others. I've talked a little bit about MDAR. That's a guideline that's supposed to be replacing actually a lot of the nature checklists and others. But you can see study participation, things like inclusion, exclusion criteria, sex as a biological variable is being asked for across all the guidelines pretty much, subject demographics, randomization, blinding, power analysis. These are all things that we know are associated with better studies. Clinical protocols, lab protocols, your protocols.io from Lenny Tettleman. I saw that he was part of this series. Are being checked by the tool. And of course, our cell line authentication statements. So we know exactly, how many authors are actually using cell line authentication to improve their papers. Roughly, this is what it does. So the author goes in, copies and pastes their methods section into the journal submission system, like at AACR. They paste that in, and then what happens is that process triggers an application programming interface to then create 3 different kinds of tables, including the SciScore report, which is intended to work with authors to go back and forth and let them know what the tool has found versus what it hasn't found. It also reformats a lot of this into the MDAR checklist, but this is a much more abbreviated thing. It's harder to find where exactly the tool had found these particular antibodies and attrition and whatever. So, and it also creates the star table for authors trying to help them do this. And of course, there's a free version for authors. So we actually got to thinking, what if we could score all papers? What would that actually look like? And so we then created the Rigor and Transparency Index. Again, this was published last year. And what this index does is it basically went through and asked the question, based on the SciScore tool, if the SciScore tool only finds 2 out of 10 of the rigor criteria in a particular paper, that paper will score a 2. If it finds 5 out of 10, it will score a 5. So what is the average score in 1990? And that answer is 2, unfortunately. And so things like description of which sex is being used went up over 20 years from about 22% usage to 38% usage. Finding sentences or finding that a paper has addressed randomization of subjects into as part of the group selection criteria went from about 80% to 30% over that same period of time. Power analysis and blinding are far behind. They're in the largely in the single-digit range here. And then, of course, the overall index, the overall rigor and transparency index went from 2 to about 4.2 over 20 years. So authors are absolutely getting better especially over this period of time from 2000 to about 2007. They had gotten better, but we still have quite a ways to go. And again, this is based on an evaluation of every PubMed Central paper that had an accessible methods section. So that ended up being 1.578 million.
Hey, Anita, you said we could jump in. So
yeah, go for it. Go for it.
This result surprises me, especially given that, a lot of the discussion and some say hysteria around reproducibility, really kind of came into its own 2013, 14, 15. But you really didn't see much of a movement in your index post those years. So that is surprising to me.
Yeah, that was a little surprising to me too. I was really looking forward to that. But this is what the data showed. I mean, you could see that big, thick black line. And there's definitely a rise here in the period of time which you'd be expecting, but it's not a jump.
Right.
And it's actually less steep than this period here. So I'm not really sure what was going on. I mean, certainly there are fewer papers, in 97, this is only a couple thousand per year. Whereas here we've got, 50,000 plus, but still this isn't as steep of a rise as I would expect.
Right. Dr. Bandrowski?
Yeah.
Beautiful work. Awesome talk so far. One of the things that I was curious about in the slide we're looking at right now is that the Rigor checklist, of course, won't apply to all studies. Like, for example, it's very relevant to animal models. But there are other studies that might not have any of these criteria in them, right?
Yes.
So what fraction of this large number of articles do you think would be very relevant to this particular checklist? And do you have other checklists? Can you sort of sort articles depending on the appropriateness of the rigor?
Yeah, and I'm actually going to start to address that in about 3 slides. But it's a great question. I mean, this, not everything can be appropriate, but I will tell you that we looked at some of these things like sex of subjects, whether it was described or not, given that they actually gave either an IRB or an IUCUC statement. So presumably if there's an IUCUC statement, sex would be applicable. And unfortunately, the numbers don't get much better. They do get a little bit better, but it, jumped from like 38% to 45%, which is, it's a jump. It's an appreciable jump, but it's not a jump that you would expect. So definitely it's better, but still not great. So we did disentangle that in this paper. Here is the data from just Nature. So these are only Nature papers from 2009 to 2019 over this 10-year period. And you can see that before they implemented the Nature checklist, which was a kind of a Herculean effort on that journal's part, you see that a lot of their, their rigor indicators are extremely low. This is definitely one of the lower journals early on here in the 2010 to 2012 range. But after this Herculean effort, they do jump to over 4.5 after a few years. So that is on the higher end of the of all the scores. It's still not as much as you might imagine, but it certainly did improve, especially randomization of subjects into groups. That seemed to go really far up. Antibody identifiability did jump too, but not nearly as much as cell. Cell went from the kind of 10% to 90%. Nature jumped from 20% to about 40%. It's a good improvement, not great. Blinding actually went up pretty well for that journal, but organism identifiability and power calculations really have not kept up yet. So, but this is what we're looking at. And again, the overall score here went from 2 to about 4.5. So, which is just reflecting, all of these individual measures. You can see kind of where, a lot of effort by the journal is having a very positive change on what's being published. But it also really shows that, there's quite a ways to go here still.
So sorry again, so
Yeah.
is the difference that you were alluding to, say, between Cell and Nature, both of which have their own checklists, is that difference then attributable to how strongly the editors and reviewers are enforcing these checklists?
Yes, it seems to be, because if you look at this, so a few slides before, I showed where the antibodies were really going up for in this period of 2015, 2016 for cell, right? This goes from nothing down here to 90%, because they're really enforcing the reagents over at cell press. But nature is really enforcing randomization, blinding, and all these other things, and those things really increase. Antibody identifiability is important for them, but not as important as it is for cell. So the different checklists do actually matter in terms of what the papers end up looking like afterwards.
That's fascinating. Thank you. Really interesting.
So if you're wondering, and people do tend to wonder whether there's any relationship with the impact factor, and the answer is no. There's absolutely no relationship. We looked at it with the raw impact factor. We looked at it in quartiles, and you can pretty much just see dots all over the place. There's absolutely no relationship in any of these. So now we're going to start to get into this question of, can we actually intervene? And can we intervene with traditional publishers? And so this is where the SciScore tool really is being used currently in the AACR literature. And people are running that tool multiple times on each paper. But can we actually do it with preprints? And in fact, we chose to work with COVID. So we formed a group around this called the Automated Screening Working Group. We've also renamed ourselves Screen It. So, this is not a company effort, because we have no idea of what to do with branding. But we basically looked at all of the COVID preprints for Rigor reproducibility criteria and many other things. This is the group of people that's involved in that. This is a good chunk of the AI community. You actually have, Amanda Cripes-Davis from ICLAC, and there are some meta researchers here, Michelle from StatCheck, and a whole bunch of others. So the tools that we're currently using to actually screen all of the COVID preprints are, of course, SciScore, which you already heard about, but there's a whole slew of others. So there's JetFighter. This is out of Caltech. And that tool is looking for non-colorblind safe pictures and graphs in papers. And what it will do is flag any papers that have color blindness compliance issues. Bazooka actually flags misleading graphs. So for example, bar graphs for continuous variables, it'll flag those kinds of things to authors. Seek and Blastin identifies some common problems with nucleotide sequences. And again, these are all basically fully automated tools. Seek and Blastin has a little bit of a review step. Trial registration screener, that just verifies that clinical trial identifiers are correct. OddPub screens for presence of open data and open code. The limitation recognizer actually recognizes and pulls out the self-acknowledged limitation sentences. So some journals ask for this, others don't. It's an important part of Rigor and reproducibility. Cite AI, this is another commercial tool. It does what's called smart citations, but for us, they're actually donating their tool to look for unacknowledged use of retracted papers, which we think is a very important piece of that authors would want to know about. And then R Transparent identifies a lot of other indicators of transparency, such as conflict of interest statements. And many more tools are starting to join. There's actually a lot of these tools that have been funded by NIH and other funding agencies. They're strewn throughout the scientific literature. They're on GitHub, which is an electronic shelf They are lovely, but they have essentially no usage after an initial paper. So my student, who actually is entering the University of California at San Diego in the fall, he's currently a high school student, has taken a lot of these things off the shelf, worked with us to put these things into a pipeline. And the pipeline that he put together basically looks for all of the preprints from bioRxiv and medRxiv that are in the COVID subset. He takes the data from that, looks at the preprint PDF, and he looks for the text. So that is processed differently for the images and then for the text. For the images, there are several tools that I listed that actually use the images to parse those. So that's that part of the pipeline. For the text components, those run through various tools. There's a study type classifier to detect whether a study is modeling or non-modeling, and we have employed a couple of other study type classifiers, but those aren't in production yet. But that will help us also trim down and ask, what is the most important criteria for this particular study? So we're really trying to work this out because one size definitely does not fit all. Thank you for that question. And so, there are some criteria that are more appropriate to some studies. We're just starting to pick that up with these classifiers. All of that is aggregated into an HTML report, and that is posted to two places, including hypothesis, and I'll show you that. And also there's a Twitter version, which is a very abbreviated report. Of course, everything gets stored in Postgres for later analysis. This is what this is what one of these things looks like. So here's a COVID preprint on medRxiv. At the bottom of it, there's a tweet that says, hey, there's a SciScore report on this, and here is a link to the report. If you click on that, you'll actually pop up this lovely page here, which says, okay, this paper with this DOI has, this sentence, which fits the Institutional Review Board statement. Here is the randomization statement from that particular paper. Here's the, where authors had addressed blinding. In this case, power analysis was not addressed by these authors, so it just says not detected, and so on and so forth. Here's an antibody. And then here are the results from some of the other tools, like limitation recognizer, which did not have a statement in this particular paper, and so on and so forth. There are currently 270 followers. I just checked this morning, but over 700,000 views of these reports. So we think that this is having some effect, hopefully, on the literature. So Nature Medicine actually agreed with us, and we were able to publish some of this. bioRxiv is actually making some additional options available for their authors to actually include and be able to be alerted about these reports. So these reports will soon be available via this this little set of links here that are being made again by bioRxiv. This is another one of the reports. In this case, nothing was detected. So, here we go. But in cases, so this will probably not be extraordinarily surprising to anyone on this call, but it turns out that preprints are not as good as papers in terms of, randomization, blinding, power analysis, or anything else. Sex as a biological variable was 19% overall. And, what you saw for 2019 in my last slides, this was about 38%. When you only look at the IRB containing papers, it's 38% that actually address sex as a biological variable. When you look at the, the comparison from at PubMed Central, the published paper, this is up in the 40s. So we're not doing as well in preprints as we are to remember all of these criteria. Seems like peer review is definitely doing something. But things like blinding and power analysis, you can see are pretty pathetic in terms of being able to address these things. And randomization, is really low for the COVID subset. So, and with that, I just like to thank all of the members of our Fair Data Informatics Lab and of course SciCrunch. And I would like to encourage all of you to add RRIDs to your next paper and of course review papers for methods. This is super important, and anything that you want to know about reagents is at rrid.site, or just type in RRID into your favorite browser and you will get to the right place. Okay, so with that, I'm hoping to take some more questions.
Yeah, we do have a few minutes for questions. We will have a roundtable, as I mentioned in your introduction, either next week or the week after. Kedar, you had a couple of questions in the chat.
Thanks, Len. Hi, Anita. That was just fantastic. Wait, let me not be rude. Let's start the video. So a couple of questions. One was, you mentioned all of this work with standardization of so many of the reagents. Are there similar authorities for chemical reagents that you use so many of?
Yeah, so there's of course the CAS system and there's PubChem. We've talked to both of them to some extent. We have not yet incorporated either one of those into the RRID initiative. If you think that, but basically what we really need is we need a willingness, because this on the part of the authorities, for the first 3-month pilot, we didn't have as much backing from some of the authorities as we would have liked. And in those cases, our compliance is okay, but it's not really great. Where we have backing from the authorities, where they're really gung-ho to try and help us, our compliance rates continue to rise. So for example, for the MMRC mice, the ability to find them has gone from, I think 30-40% all the way up to 95%. It's right now, it's really quite good, and they've been really good at supporting the initiative.
And so does the push come from, so for example, PubChem itself, or from the manufacturers? Do they essentially do some arm twisting to get the manufacturers to have UIDs?
Yeah, so we really need both. And for the antibody registry, there is a lot of antibody manufacturers that are reflecting now the RRID right on their website, which is great, because that's where people go. They don't necessarily go to the antibody registry. They're going to Thermo Fisher. And if Thermo Fisher has a big box that says, click here for proper citation, and you can copy that, that's going to be a lot of traffic and a lot of eyeballs that are seeing that, saying, oh, this is how I'm supposed to cite this thing at the place where I go and buy it. Cellosaurus, for cell lines, we haven't been as successful getting those IDs into the manufacturers. And while, that part has been very successful, we have thousands now, many thousands of cell line RRIDs, it's not as successful as the antibodies. And so I think you kind of need partners on both sides of that commercial, non-commercial realm to really kind of hold hands and say, all right, authors, let's corral you, just a little bit, and then to really improve the whole situation.
Dr. Bandrowski, this conversation is kind of moving towards a question that I had, which was, it seems like there's a major opportunity here for vendors really to highlight their products by demonstrating, hey, our products are being used with, outstanding research studies. And this is why you should want to choose our reagent over someone else's. And are you seeing that kind of response by the vendors?
Yes, we definitely are, especially in the antibody space, which, again, I run the antibody registry, so I see it day to day. We're starting to see it out of instrument manufacturers as well, but that's much more of a nascent effort. Which again, we can talk to those instrument manufacturers, and they're looking at it and saying, okay, we want to know where our stuff is being used. Whereas again, ATCC has not yet come around. So we're at different phases in with different kind of manufacturing areas, right? And different kinds of reagent manufacturers or organism stock centers. So what I'd love to be able to do is just continue to move in the right direction with a lot of those. So any help there, any thoughts, I would love it. If you had any additional pull with, say, an ATCC or any of these guys, and certainly making sure that anything on your end has RRIDs would be hopefully theoretically a no-brainer, and it would be great for these manufacturers to also start seeing that around.
Definitely.
Hi, this is Ethan Nemetrovsky. I just wanted to make a comment as an end user. My last two papers published a couple months ago in AACR journals, I got a SciScore, and I had no idea what it meant. And I had to go online to kind of look and see what the score meant. I was going to suggest as an end user that you, in fact, Len, I think I even asked you what a SciScore was, if you remember. Yeah, I had no idea what a good score was, wasn't a good score, or what to do to improve the score. So I'm just suggesting, at least for ACR, and I'm on a few ACR editorial boards, among other journals, and I just didn't know what to do with the data. I wanted to do the right thing, but I didn't know what the right thing was. So just wanted to tell you that the tool looks really informative now that I heard the talk, but I had no idea what it was when I received my score.
Okay, well, that's really good to know, and we'll try to, I'm working pretty closely with AACR to try and figure out, how to get people to do a better job, and the information around SciScore has definitely been improving over time, but I think we're not quite there yet. So thank you for that feedback. I really appreciate it.
One of the things that what I did is I went to Google SciScore, and there wasn't an authoritative site that explained how to interpret. See, that's, as an end user, that's what I wanted to know, kind of why it wasn't perfect. How do I get, I'm a scientist, I want to do things well. And so I just wanted to tell you, it's not a criticism, it's just an end user observation. And I really enjoyed your presentation. I just want to apologize. I have to run to another call.
No, that's fine. I understand. And we'll pick this up, I think, next week. So
yeah, I was going to say, Anita, there are definitely a couple of things we should pick up, including your experience with ATCC, which I think I could be potentially helpful with, given my previous relationship with them. But the very last question I wanted to ask, which is sort of related to exactly what you were just talking about, is, you've spent a lot, appropriately so, spent a lot of time working with journals and with vendors. What about funders? And the big 800-pound gorilla in the room here is, who. What's your experience been, with NIH and other funders?
So we're having some conversations with some of the metrics pulling, parts of the 500-pound gorilla, which have been very interesting. I don't think we're quite there yet. We have talked to some of the wonderful people over at in review, and they basically told us to go away because there's like a 15-year turnaround for any change to any of that. Right. So we'll see. We actually have worked with Simon's Foundation, that a lot of these smaller funders are much more nimble, and so we were able to pull SciScores for their potential grantees from multiple papers and kind of say, okay, well, it's nice that this person is saying that they'll use blinding, but they actually didn't use blinding in their last 5 papers. So it's something that can be a potential lever for something like the 500-pound gorilla and maybe some other funders too. But you know, we now, because we have these scores and we're actually running a whole other set for, you know, for the 2020 papers, what we can know that, right? We can know who has a better, you know, I don't know if it's an H factor or an RT factor, or I don't know. But that's definitely something that we'd love to talk to anyone with any knowledge of the 500-pound gorilla.
Well, thank you so much. This was it was a terrific talk and a really great summary of all the work you've done over the years on RRIDs, and we really look forward to following up with the roundtable next week. So thank you again, and enjoy the rest of your time in the Big Apple. And we're looking forward to again following up with you.
Great. Thank you very much.
Thank you, Anita.
Thank you.